Continue on with Chapter 2, the chemical organization. We are going to look at nucleic acids. Nucleic acids are what make up your entire genetic code, which is stored in DNA, and also a type of molecule called RNA, which helps read the genetic code and translate it into protein synthesis. So, nucleic acids are large organic molecules. Typically, we will find most of them in the nucleus. And the nucleus is the storehouse, the nucleus of the cell, which we'll see in Chapter 3, is the storehouse of your genetic code. And the genetic code is made up of deoxyribonucleic acid, and abbreviated DNA. We've all heard of DNA, and it is made up of what we call nucleotides. And we also have a different variant, it's quite similar, but it's a little bit different, called ribonucleic acid, or RNA, which makes up smaller molecules that go in and out of the nucleus of the cell and help translate the message of DNA, or I should say the blueprint of DNA, which contains your genetic code, into protein synthesis. So DNA determines your inherited characteristics, i.e. how tall you can become, how strong you can become, what color your eyes are, what color your hair is, and that sort of thing. Of course, there's environmental factors that determine this as well, but the parameters of your potential development are all contained within your DNA. It directs protein synthesis in the body, and enzymes are proteins, and it also helps control metabolism because metabolism is controlled through enzymes. So, the structure of DNA and RNA, they're made up of things called nucleic acids. Now, remember when we talked about proteins? Proteins are made up of amino acids, and these amino acids are strung together in large chains or long chains called peptides, and when they get really long, they're called, or I should say, polypeptides, and when they get very long, they become proteins. And this is what we call polymerization, because they have functional groups that we talked about, an amino end group on one end and a carboxyl group on the other end, and these things link together through dehydration synthesis. We'll see something similar, but with different functional groups in nucleotides. And nucleotides are basically what we call the building blocks of DNA and RNA. And they also have very distinct entities that make up the molecule. So you have three molecular parts, a 5-carbon sugar called a pentose sugar. It's, in the case of DNA, a deoxyribose sugar, or a ribose sugar in the case of RNA. We're not really going to differentiate between the two. Suffice it to say that deoxyribose is part of DNA and ribose is part of RNA, and hence the abbreviations. So you have a sugar. It's a 5-carbon sugar. We'll look at it in just a second. And it makes a ring. And the ring looks kind of, it's got 5 corners to it. Looks like a pentagon, so that will help you remember that it is a pentose sugar, or we call it a ribose sugar. And it has a phosphate group on one end. And it has something sticking out of its side called a nitrogenous base. And there are 5 of these that we'll look at, 4 of which are in DNA, adenine, guanine, thymine, cytosine, and 3 of these, adenine, guanine, and cytosine, are in RNA, but replacing thymine and RNA, we have uracil. And we'll look at what those look like in just a moment. So here is the basic nucleotide structure. Here we have your 5-carbon sugar. You've got a carbon here, you've got a carbon on each of these corners here, and then you've got an oxygen at this corner. Now, the reason when we write these little ring structures, if there's a carbon at the corner, we don't even write it in. It is just understood that there is a carbon there. Here we have a hydroxyl group. You should recognize this as one of your functional groups. And on this end, we have a phosphate group. And as you should recognize, a phosphate group is one of your functional groups. And this part is called the nitrogenous base. You have adenine, guanine, cytosine, thymine, or in the case of DNA, uracil replaces thymine. And what you will have is you will have linkages of these things together in long chains called nucleic acids. So the phosphate group will link through dehydration synthesis to the hydroxyl group, and water will be a byproduct of this reaction, and you'll have long chains of these things. Now, depending on whether you are In DNA or RNA, they will be double-stranded or single-stranded respectively. And we'll look at that in just a little bit. So here are two types of nucleic acids. You see adenine here and guanine here. Both of these are examples of what we call purines. And purines have a nitrogenous base that has this double ring structure here. So you have two rings. There are lots of nitrogen in them, hence they're called nitrogenous bases. And each of these purines have these two ring structure. On the other hand, you have something called pyrimidines, which have a single ring structure. And here we have cytosine and thymine. Thymine is what we find in DNA, and uracil is very similar to it, and it's found only in RNA. So when we go into chapter 3, we'll see how RNA will sort of copy the information in DNA and relay the message that is stored in DNA and then help translate it into protein. We'll look at that in a lot of detail in chapter 3, so I don't want to go into too much detail just yet. Suffice it to say that uracil is found in RNA, where thymine is found in DNA. So, as we mentioned before, DNA is double-stranded. So what happens is these bases will make hydrogen bonds with one another. And you can see hydrogens and how they could make hydrogen bonds with one another. And in DNA, the hydrogen bonds will hold together. Now, you've probably seen DNA. You probably all know that it has a double helical structure. And it looks kind of like a staircase. And the rungs of the staircase are formed by these nitrogenous bases that are being held together with these hydrogen bonds. Now, going back to our nucleic acids here, we can look at our ribose sugar and our phosphate group. These are the things that link together to make the long chain. So basically what you're going to have is what we call a sugar phosphate backbone, because you have lots of these things linking together and these things sticking out the side. In DNA, these are the things that hold together with the hydrogen bonds to form this helical staircase. However, RNA is single-stranded, and you just have this sugar phosphate chain that has these nitrogenous bases sticking out of the side of it. Now we have in DNA what we call complementary base pairs. So purines always pair with pyrimidines, and that's a size issue. You can see that if you had two of these, they wouldn't really fit in the space between the helix of the DNA. So you have always purines pairing with pyrimidines, but more specifically, you have adenine always pairing with thymine and guanine always pairing with cytosine. And in the case when you have DNA and RNA interactions, what you're going to see is adenine will pair with uracil because uracil is an RNA. All right, so here are our base pairings here, adenine with thymine, cytosine and guanine, and in RNA, uracil replaces thymine. I always remember this as forms AT, and CG or GC, you can think of it either way. I always think of that as Greensboro College. So if you're at Greensboro College, you can always remember how base pairs. And you can also always remember that uracil replaces thymine when we're talking about interactions between DNA and RNA. And we'll see how that works in chapter 3. All right, so here is that double helix structure that I was talking about. Here is this sugar phosphate backbone, because here is your ribose sugar right here. It has this phosphate group up here. Here we had a hydroxyl group, and through dehydration synthesis, it linked with this phosphate group from another ribose sugar. And each one of these things have these little nitrogenous bases sticking out the side. You can see how they're hydrogen bonding here. And these are all covalent bonds. So in this sugar phosphate backbone, these are all covalent bonds. And this is abbreviated by this sort of beige line here, this beige slab. And each one of these represents one of these nitrogenous bases. So here we have like a small strand of small nucleic acid. But here we have DNA, and we have a portion of it here. Obviously, DNAs are hundreds and thousands of base pairs long. And we can see how we have these hydrogen bond interactions here between adenine, thymine, guanine, cytosine. This is DNA. And we have this thing, which is this thing wrapped around like this. And there are two of them, and they're forming this helix, the helical staircase, with the nitrogenous bases being like the rungs or the stair steps between the two sugar phosphate backbones. And here we can see how adenine pairs with thymine and guanine pairs with cytosine. And you can see that this is a purine and this is a pyrimidine. This has the two-ring structure and this has the single ring structure. All right, so we have RNA, which is very important as well. RNA is typically in a single strand, and we have three types that we're going to look at in a lot more detail in the next chapter. We have one called messenger RNA, one called transfer RNA, and one called ribosomal RNA. And when we look at functions of the cell and how it makes proteins, we're going to look at all of these in a lot more detail. So in addition to storing the genetic code and being used to make proteins, nucleotides are also very important for energy storage. One of the most common ones that we will see is adenosine. And adenosine is basically an adenine, an adenosine, which is a ribose sugar, a phosphate group, and an adenine nitrogenous base sticking out the side of it. But instead of having one phosphate, adenosine diphosphate has two. The second phosphate bond is quite energy. It's full of energy. So it's a good way to store energy. However, adenosine triphosphate has even more energy. And for each phosphate that you add to this thing, you're adding energy. So this phosphate is added through a process called phosphorylation, which, not surprising, is adding a phosphate to something. In this case, we're adding a phosphate to ADP with a high energy bond forms ATP. This is the highest energy bond that we're going to see in this type of construct. So ATP is a very common energy source. We'll see it in muscle. When we study the muscle chapter, we'll see it a lot in that chapter. And adenosine triphosphate is this high energy molecule, ATP. And ADP actually has some energy in it as well, but not as much as ATP. And it just has to do with how many phosphate groups are on that molecule. We'll look at that molecule in just a second. Needless to say that if you can put phosphates on it, you can also take phosphates off. So if you catalyze, that is, break the last phosphate group off of adenosine triphosphate to get adenosine diphosphate, then there's an enzyme called adenosine triphosphatase that does this work, or cleaves this phosphate off of the ATP to make ADP. Anytime you see the ending ase on something, we're talking about an enzyme. So remember that. Ase means enzyme. ATPase is an enzyme. All right, so here is the actual molecule. Here's our adenine, here's our adenosine, here's our ribose sugar right here. Here we have our first phosphate group. Here, this would be ADP because we have two phosphate groups now, and this is a high energy bond. And the third phosphate group has an even higher energy to it. And so this is a way of storing energy. As we talk about energy and order a little bit later in this chapter, we'll talk a little bit about how the terms for making and breaking these bonds and the kind of energy that's stored and the kind of work that can be done. So that brings us to energy and order, because we have several types of reactions in the body that can either help us store energy or can be accomplished to release energy. So energy, what is energy? First of all, it's the power to do work. And there are several types of energy that we need to think about, one of which is simply kinetic energy. This is the energy you see when something is in motion. If somebody is running, if a car is moving, or an airplane is flying, that is an example of kinetic energy because it's moving. Potential energy is simply the stored energy that will allow kinetic energy, when released, when the stored energy is released, it will allow kinetic energy to happen. And the common example that we see is if I take a book and hold it up in the air, hold it up over my head. That book has potential energy because it is off the ground, and if I drop it, gravity will pull it towards the earth. And so that book, while I'm holding up over my head, has potential energy. It's not moving, but it has the potential to do so. As soon as I drop the book, then as it falls to the earth, it has kinetic energy. Now, chemical energy is basically a type of potential energy, but it's stored in chemical bonds. It's stored, for example, in the chemical bonds that we see in ATP. That last phosphate group on there has a high energy bond. And so there is energy. Once you cleave that bond, that can then be used to do work. Very similar to the way gasoline can be used to make your car engine run. Gasoline has stored chemical energy. Batteries have stored chemical energy. So it's a chemical energy that is potential energy that once it's released, it can do work. And work itself is a change in mass or distance, and it's defined as work equals force times distance. And it can also be used to describe an internal change in a molecule, an actual reorganization of the molecular structure. Work has to happen in order to do that. And that brings us really to chemical reactions. So chemical reactions are where new bonds are formed or existing bonds are broken. Now, we went over the types of reactions a little bit earlier in the last class, and we'll probably touch on them again as we move through this section of this chapter. But you have, in a chemical reaction, you have the things that are going into the reaction. These are the reactants. The products are the things that come out of the reaction. So if you're going to make a product and you put the components of that product together, the components are the reactants and the product is the final end thing that you are making. So, for example, we talked about these chemical reactions earlier. We have a decomposition reaction, and that is where you take two things and break them apart. In this case, the reactant is AB. The products are A plus B. Now, if we're doing a synthesis reaction, and I'll talk about anabolism and catabolism in just a second. If we are doing a synthesis reaction, then the reactants are A and B, and the product is AB. Now, we talked a little bit about the kinds of reactions that we see a lot in this course, and a good example of a decomposition reaction is hydrolysis, because we're using water to break a molecule. So we have AB, and we break it apart by adding water, and then you have hydrogen becoming part of A or B, in this case it's A, and your hydroxyl group becoming part of B. So when you have a dehydration synthesis, which is the opposite of hydrolysis, and we talked about this a lot in the preceding lecture, what you do is you're taking water out of the reaction. So you have a hydrogen, in this case, on this molecule A, and you have a hydroxyl group on this molecule B, and these are the reactants, and the product is AB, and water is your byproduct. So you could say water is a product too, but in this case, we're mostly interested in AB and water because that's not the main thing that we're forming as a byproduct of that reaction. So I'll talk a little bit about what catabolic reactions are and anabolic reactions as well. Catabolic reactions are things that break things down. And you can sort of think of catastrophe. If something has a catastrophic failure, it breaks into little pieces, right? A synthesis reaction is something that put things together, and it's anabolic. Anabolic is building up. So literally, catabolism means a throwing down of something. can also think of catabolism, breaking things apart, as if you have cats in your house, like I do, and I come home at night and there is stuff everywhere because the cats, there have been a catastrophe and they've torn everything down in my absence. And it's not surprising for me to find the mail that I took out of the mailbox and put neatly into piles on the table all over the floor when I get home. So you can think of catabolism as breaking down. And anabolism can be kind of easy to remember because it's a synthesis of things. And in the body, when we synthesize things, we build up. Like when we synthesize proteins, proteins are a major component of muscles. So we build muscles. And it's true that some unscrupulous bodybuilders will use anabolic steroids. Anabolic means building up. So we talked about the exchange and reversible reactions as well. And an exchange reaction simply involves first a decomposition and then a synthesis reaction. This is simply where you take reactants made up, say, of two components each, in this case A and B, and C and D, and then we just exchange the components. So A now has D, and C now is associated with B. So we have the products AD and CB. And as we've said earlier, a reversible reaction is something that can go in either direction. So in the case of, say, an anabolic reaction where we have A plus B to get the product AB, we can also have AB being broken down, and thus AB becomes the reactant, and A plus B, or A and B, are the products, because it's going in both directions. Now, when a reaction reaches equilibrium, then this is happening at the same rate. So for every reactant we have on this side, where we have A getting together with B to make AB, somewhere else a molecule of AB is breaking down into A and B. So what we have here, it's really hard to say which are the reactants and which are the products because they're in equilibrium, but we actually have, you could say AB is a product of the reaction A plus B, and at the same time, in equal numbers, you have AB being a reactant that is breaking down to form products A plus B. So it just depends on the direction of the reaction, and when the reaction is happening in both directions equally, it's called equilibrium. And this is sort of what's happening when you go back to our pH example of water, where water will spontaneously break down into hydrogen ion and a hydroxyl group. And it's approximately 1 and 10 to the negative 7 water molecules in a mole. So when that happens, then you have this tiny amount of hydroxide ion in water. But as many water molecules are spontaneously breaking down, you have some of these hydroxide ions reassociating with hydroxyl ions to form water again. So this is what we mean by a reversible reaction. It's happening at the same rate in both directions. And that's why water doesn't spontaneously become basic, nor does it spontaneously become more acidic, because you're not going to have more spontaneous breakdown of water into hydroxide ion and hydroxyl ion, then you're going to have a reassociation. It's happening at exactly the same rate in both directions. And that's a good example of what we mean by equilibrium. All right. Now, when we break things down, this is what's called an exergonic reaction because it produces energy. And that would be an example of when we break that third phosphate off of ATP. It is used to create energy that can then be used by, say, muscles or something to do work. And we'll see a lot of reactions in the body that use ATP for fuel. Now, exergonic means energy going out. Exer, going out of, and gonic meaning energy or having to do with energy. We find that exergonic reactions are also exothermic reactions because they generate heat. So you have an outflow of heat. So whenever you think about when you're doing a lot of work and your muscles are really working hard, what happens? You heat up, right? That's because exergonic reactions are exothermic reactions. Now, endergonic reactions are the opposite. These are reactions that are used to store energy. So they use more energy than they produce. And so by putting energy into these things, these molecules that we're building, we are now storing energy. They also tend to take heat out of their surroundings because you're putting the energy into the molecule itself. For example, in ATP, when you make adenosine triphosphate, you are adding that third phosphate to adenosine diphosphate, and creates a very high energy bond. It took energy to make that bond, but now that you have that phosphate attached, that third phosphate, you have stored energy. And so later that bond can be broken and used to do work. So that brings us to the whole idea of catabolism and anabolism again. Catabolism, as we said, literally means a throwing down. So it's breakdown of larger components or compounds into smaller molecules or even individual atoms, and this releases energy. So when we take that third phosphate group off adenosine triphosphate, that is catabolism. It releases energy. When we put that third phosphate back on to adenosine diphosphate to create adenosine triphosphate, that is anabolism. That's where we're creating larger compounds from smaller molecules or individual atoms, and it requires energy to do so. As I've said earlier, anabolism is like a building up. So when you build your muscle, it takes energy to do this. This is why bodybuilders and athletes have to eat a lot of food, because they're expending a lot of energy and they're doing a lot of work. But bodybuilders are building muscle, and it takes energy to build that muscle. Now, metabolism is basically the term we use to describe all the chemical reactions that are going on within an organism. It is used to encompass all catabolic and anabolic reactions in the body. So, chemical reactions in the body are quite interesting because usually we need something to get them started. All chemical reactions require an activation energy, and that is an amount of energy that is required to start a reaction. A lot of reactions just don't start spontaneously. So things that we want to do in the body, we can't just expect them to happen. They have to be helped along. And this is why we have enzymes. Enzymes are basically catalysts. That's what they do. And what do catalysts do? They help reactions happen. You can kind of think of them as like a matchmaking service. So you've got your molecule A that you want to join with molecule B, and molecule A and B are just floating around out there, and they may or may not bump into each other on their own. But an enzyme will help them come together and meet and then make that bond. And they do this, they lower the activation energy of a chemical reaction. And we'll look at a little graph that shows this in just a little bit. But what's important is they themselves are not changed in any way. They do not become part of the reaction. They are not changed, and they're not used up in the reaction. So when you have an enzyme and it's bringing together these molecules to join together, then once the molecules are joined together, the enzyme releases them, releases the product, and then it looks for more molecules to join together. So there are some things about enzymes that we need to understand. The enzymes work specifically with certain types of molecules or atoms that they're trying to bring together. And these atoms are called substrates. The substrates are what the enzyme puts together. So in my example, when I said we had a molecule A and molecule B, molecule A and molecule B are substrates of the enzyme. When the enzyme gets them to bond together, then it releases them, and then it can go work on other substrates. But the substrates are always going to be A and B. It's not going to go off looking for C and D molecules to put together. So this is what we mean by specificity. So when we have an ATPase, for an example, ATPase is specific to ATP, and what it does is it cleaves that third phosphate group off ATP to make ADP. It doesn't do other types of reactions. That's what it does. Now, enzymes also have saturation limits, and that means that, well, for one thing, they can only do so many catalytic, I don't want to say catalytic conversions, but kind of funny, but they can only catalyze so many reactions per second. So if you have an enzyme, say, that can catalyze 1,000 reactions per second, and you have 100 of them in there, then they can only catalyze 100,000 reactions per second. The only way that you can increase the speed of reaction is by adding more enzymes, pretty much, because once they reach their limit and there are enough molecules in there that they're trying to catalyze a reaction between, then you can actually have more molecules than the enzyme can convert into the products that it's going to release. So that's what we mean by the saturation limits. These things become saturated when they can't catalyze any more reactions. They're already working at maximum speed. As I say, the only way to increase the speed of reaction is by increasing the number of enzymes, of enzyme molecules, I should say. All right, so these enzymes can be regulated by other molecules or chemicals, and we'll see this in a little bit. We have cofactors and we have coenzymes that might be necessary to turn on an enzyme, and I can show that a little bit later on. Right now, suffice it to say that these enzymes, that when we went over protein structure, you have globular proteins. These enzymes are globular proteins, and they have little pockets or areas that their substrates fit into that help them pull these substrates, or I should say push these substrates together. And that particular conformation that it has, its quaternary structure or tertiary structure, if it's just a single polypeptide or a single protein, that can be changed. The shape of that thing can be changed when it binds to other things like cofactors and coenzymes, as we'll see in a little bit. And if those things don't bind, then the enzyme isn't on. So sometimes you have to have like a little key or something to turn on the enzyme. So here's an example of a reaction that's catalyzed by an enzyme. Here we have reactants, and normally they would have to have a big activation energy to get them to come together. So, for example, if you think of a bunch of molecules floating around in air, and they may or may not react with one another if they encounter each other kind of randomly. Now imagine you have these molecules within a container, and they're floating around, and they react with one another whenever they encounter one another. And that may not be very often. But if you heat that container up and all the air inside that has these molecules in it starts moving faster and faster and faster, then the reactants might bump into each other a little bit faster and encounter each other more often and be more likely to make bonds with one another. Now, that's a good example of an activation energy. The reaction has to take place because there's some kind of energy that was there to make the encounter of these molecules possible, or make them in some way interact with one another to form a bond. So without an enzyme, you usually need a fairly high activation energy. In the body, we may not have that option. For example, if you want to break apart sugar, like glucose or something, in the laboratory, you pretty much have to put it in acid and boil it. Now, our bodies can't do that. That would destroy our tissues. So that's why we have enzymes. Enzymes lower this activation energy substantially, and they help these molecules, as I say, like a matchmaker, goes around looking for appropriate molecules to pair together, or in some cases, to break apart molecules that otherwise you would need a lot of energy to do that. So what the enzymes do is it basically lowers this activation energy into a reasonable amount that can happen within the body without adding too much heat. And that way you can get stable products without having to heat these reactants up to super high degrees or expose them to really toxic or highly caustic, high pH, or anything like that. So here is an example of an enzyme. It's kind of this, here's the cartoon version, but we have this globular protein here, and you've got these molecules here that it wants to bring together. All right, so it's the matchmaker, it's looking for these things, and here's a substrate. We'll just call it S1 and S2 to represent the substrate 1, some molecule, and substrate 2, another molecule that is specific to this enzyme. So notice these things have these little pockets. The enzyme has these little pockets that fit with part of the substrates. And so the substrates are then sort of pulled into these pockets by the enzyme, and then here's where the substrates would bind with one another. And so the substrates are then induced to bind with one another. And once they've done so and they've made this product, then the enzyme can release it and it's ready to catalyze another reaction. Now, this can be used anabolically to build products, to build a single product from multiple substrates, or you can have one molecule that is a substrate that is then broken down. So enzymes can either build molecules or break molecules. But a lot of these reactions that occur in the body are reversible. So when you break something, when you build something up, for example, when you make ATP to store energy, later you'll want to break it down so you can get the energy back out of it. Enzymes are responsible in both cases. Interestingly, they are different enzymes. A different enzyme will build up ATP, then will break it down. So ATPase, which breaks it into ADP and phosphate, is a different enzyme from the one used to build ATP. So as I mentioned earlier, you have these things called cofactors and coenzymes, and both of these things are things that kind of turn the molecule on. So some enzymes require them to function. So if you don't turn the enzyme on, it won't work. And this is one way of regulating it within the body. So sometimes you want an enzyme to be working. Other times you can turn it off because you don't need it. You have enough product and you don't need any more. So you have something called cofactors. And this can be like an ion or small molecule that binds to an enzyme before any substrates can be bound. So it actually changes the shape of it a little bit. So the cofactor, without the cofactor, it's not in the right shape to bind the substrates. Now, with the cofactor, you've turned it on and you've made the molecule take that shape that's going to allow it to bind with these substrates and cause them to bind together. Or, in the case of breaking down, it's going to be in the right shape to break a molecule. And a coenzyme is very similar to a cofactor. It's just a non-protein organic thing, such as vitamins, like vitamin C, for example. And we'll look at those a little bit later on. Now, a lot of times you have multiple enzymes. They're different. They're different proteins. They may be very similar, but they catalyze the same reaction. So sometimes in some tissues, you'll have an enzyme that catalyzes a reaction, and in some other tissue, the enzyme catalyzes the same reaction, but it's a slightly different enzyme. Does the same thing, but it's kind of like if you have two Toyotas, let's say you have a Prius and a Camry. Maybe that's not a great example, but they're the same. You use them to drive somewhere. So they're cars and they serve the same function, but they're slightly different. And you may find this from organism to organism and even within an organism from tissue to tissue. So these are called isozymes, meaning the same. So they're enzymes that do the same thing, but it's kind of like anytime you see the word iso, you think of the same. So as we discussed last time, or we touched on it last time, the enzymes have an optimal temperature and an optimal pH in which to work. And if you get them outside of that, because we looked at the protein structure that makes up enzymes, if you put it in a pH that is out of its normal operating range, it'll start to denature. And if you heat it up super high to high temperatures, it can denature. And what that means is that the structure, the three-dimensional structure, be it the quaternary or the tertiary structure, the three-dimensional structure of the protein starts to break down. And eventually, these things can get so hot if you heat them up that they'll break down into their individual amino acids. And then you just have a bunch of amino acids. But when you start to destroy the structure of a protein, this is called denaturation. And this is the loss of the shape and hence the function because you've changed the heat or the pH. In our case, it's usually when you increase the heat, like when you boil, let's say you fry an egg, and you look at how the egg looks before you fry it, and then you fry it, and you get this white and yellow mass as the proteins denature and just form this kind of basically mass of stuff. All right, so that concludes chapter 2, and the next lecture that I will give, I'll plan to post one more video lecture, and it will be chapter 3, the cell. And until then, we'll see you on Tuesday.